have to say it's quite a bit intimidating after this distinguished speaker to present my own research, but I will give my best. So I was invited to talk about precision fermentation, and I focus my presentation on milk and milk products, but you can relate the technology also to human milk, actually, so it's not that different. But before I start, I want to quickly give you an overview. What is precision fermentation? What am I talking about? So David already nicely introduced it very well. We work with acellular products, so we still start with a cell, a single cell. So it can be a yeast, a fungi, a bacterium. It is genetically modified to introduce the target protein we are aiming for. For example, a milk protein can be a human milk protein or a bovine one. We put it in a bioreactor at perfect growing conditions, and then we use them as cell factories. So after stimulating them, they start secreting our target protein. And since the proteins are much smaller than our single-cell organisms, we can more or less capture them in a membrane and then just isolate them, have the proteins like a protein isolate, and just the GMO, the genetically modified organism, is not part of our food. So we are relatively safe there. It's only novel food. It's not genetically modified food. The proteins we receive, this technology has been already used 40 years ago for insulin production. Insulin is a peptide hormone. So if someone has diabetes, they certainly have a protein that is done by precision fermentation. But nowadays, it has become more affordable, so we can also use it for producing milk or egg proteins or gelatin or whatever else you can think of. But let's have a closer look at the proteins we actually produce, because as a take-home message, I will tell you that the proteins we are producing are not perfect copies of what you would think of the target protein. So this is just one of our studies with milk protein. It's a whey protein, beta-lactoglobulin. This is well characterized, so we always start with that one because we know everything about this protein. This is the amino acid sequence in one letter code. We know that it has 162 amino acids. And when we start copying this protein in our cell factories, and this was yeast, the other was a bacteria, then you see you don't get the same sequence. So we always had some minor amino acid mutations, highlighted in yellow here. I don't want to go into too much details, but there are minor changes, and these might affect how the protein behaves in a food product. For example, it could affect allergenicity, it could affect the digestibility, and it could affect the functionality. So we need to test all these parameters for each individual protein we are producing, each host we are using to produce the protein, and the fermentation conditions, and so on. So it's quite laborious, as you can imagine. Since I'm from food process engineering, I just quickly want to highlight the part of the functionality. As you might wonder, okay, 1 or 2 amino acids exchanged, what does it do? Well, we want to make a food product, ideally, out of our protein. So here we did a kind of competition, animal protein versus the cell factory protein. And in this case, we were able to have a similar temperature behavior, similar solubility, similar creaming stability, and we were able to make a milk drink out of it. So you'd say these minor mutations didn't really do much. We were able to make a nice drink. Of course, I didn't taste it. It's not allowed in Europe. So it just looked nice. It behaved nice. But yeah, we have to test further. And then, due to the time, it's not much, I can delve into not much details, but I also want to have a look on the caseins. And here is a nice link to the previous talks, because caseins are actually the proteins that mediate the nutritious quality of milk. So as you can imagine, milk is the only product that has gone through an evolution to be as nutritious as possible by nature, as it is intended to nourish a growing mammal. And in this case, the caseins assemble into a micelle. You can see this ball here. It's a protein ball of 200 nanometers. And it has quite an intricate structures of kappa caseins on the outside, alpha beta caseins in the inside, and calcium phosphate clusters. And the reason is if you ingest it, it coagulates in the stomach. It introduces satiety. by being very viscous and slowly releasing the calcium. It would otherwise be insoluble. So this calcium density is only possible in that structure. And next to that, from an engineering perspective, also this structure is needed for making cheese. So what we do is, in our bioreactors, each of the caseins is produced individually. Then we try to reassemble it into nature's structure of forming a micelle. Unfortunately, the slight structural modifications affect our assembly, so we get misshaped micelles. And our most recent publication about it, you can see that there are parts necessary to form a cheese, so we're a bit off yet. So we still need to work on understanding better how to assemble structurally different caseins, or to improve our caseins. And with that, I would like to close. I was talking about proteins and structures from proteins, but milk is way more complex. So if a cow produces milk, you have lipids, you have lactose, you have vitamins, you have minerals, and so on, probiotic effects, and proteins are just part of this. If we use our bioreactors to produce proteins, we just have, I often say we have Lego bricks. We need to assemble all the Lego bricks back to the one that is resembling milk. So we need to also choose the fat, the lactose, the vitamins, and so on. It gives nice opportunities to understand better how milk is made of, but we could also, in theory, produce a better milk if we understand how it works. And finally, this is a multidisciplinary effort. So I cannot do this on my own. So we have people working on safety, on regulative issues, on sustainability of the whole process. And I think together we will be able to connect the next step in this research. And with that, I would like to close. I'd like to thank my research group that did the actual work. So I'm just presenting it. And thank you for listening.